Okay, so for this video, we're going to review the remaining two steps of aerobic cellular respiration, the Krebs cycle and the electron transport chain. So first, you have kind of this pre-Krebs cycle leading from between glycolysis and the Krebs cycle. It's called various things. I usually call it modification of pyruvate. Sometimes I've seen it called grooming of the pyruvate. It's just getting the pyruvate ready for the Krebs cycle. So this occurs as the pyruvic acid passes through the two membranes of the mitochondria and into the matrix of the mitochondria. So we can see here's our pyruvic acid. It's going through the outer membrane. Now it's going through the inner membrane. And then here's the process of the modification of the pyruvate. So what you're going to do is break down the pyruvic acid a little bit more and get ready for the Krebs cycle. And so what you end up doing is changing this 3-carbon pyruvic acid or pyruvate into a 2-carbon molecule called acetyl-CoA. Since you have lost a carbon, you're going to release that as carbon dioxide. And since you've broken a carbon bond, you can also produce a little bit of energy. You can store some high energy electrons in NADH, one of our electron carriers. Okay. Now, we call this a molecule here acetyl-CoA, but really the 2-carbon group is the acetyl group, and the CoA is just an enzyme that comes in to help transport the acetyl group into the Krebs cycle. So the acetyl is the 2-carbon, the CoA, so this whole thing is acetyl-CoA. Once it delivers the acetyl group into the Krebs cycle, this CoA will go off and do this again and grab another acetyl group. Okay. So as we said before, CO2 and NADH are produced in this process when you go from pyruvate to the acetyl group, which then joins with the acetyl-CoA to enter the Krebs cycle. Okay. So the Krebs cycle, also called the citric acid cycle, because that is the first molecule formed. So this occurs in the mitochondrial matrix, as we saw in the last picture. And you're just going to continue to break down the glucose to produce ATP and a lot of electron carriers. In a way, you can think of it as the opposite of the Calvin cycle in photosynthesis, because instead of adding in CO2, you are pulling off CO2. Instead of adding in electron carriers and ATP, you're getting electron carriers and ATP out of this process. So the first part of this, as we said, is to add in the acetyl group. So there's a 4-carbon molecule that's left in this cycle floating around the mitochondrial matrix. It is going to combine with that acetyl group, and you're left with this 6-carbon citric acid. So that's kind of the first step here. Oh, and there's the information on the slide. So acetyl-CoA adds the 2-carbon acetyl group to a 4-carbon molecule already present, producing a 6-carbon molecule called citric acid. Hence why one of the names of this is the citric acid cycle. Okay. So now we're going to try and get energy out of this 6-carbon molecule. So that citric acid is going to be broken down into a 4-carbon molecule again, and more CO2 is going to be released. So you got your 6-carbon, you're going to break off a carbon, release it as CO2. When you do that, you can produce some NADH, and you're left with a 5-carbon molecule. You're going to break off another CO2, you get some energy out of that, store it as NADH, and you're left with a 5-carbon molecule. Or excuse me, a 4-carbon molecule. So again, citric acid, 6 carbons, you change it into a slightly different 6-carbon molecule, remove a CO2, release energy. You're left with a 5-carbon molecule, remove some carbon, release some energy, and you're back to a 4-carbon molecule, though not the 4-carbon molecule that you started with. Oh, sorry, let me go back. I thought I had something else. So at that point, once you have that 4-carbon molecule, you want to change, got a bell going off, so I got to quickly finish this. You're going to change that 4-carbon molecule around and regenerate back to the original 4-carbon molecule. So in order to do that, or when you do that, you can get some more energy out of it. So we can get an actual ATP. We can get another electron carrier called FADH2. Has a little bit less energy, but it can still store some high energy electrons. We got some intermediates again, and then finally getting back to our final intermediate, that 4-carbon molecule that we started with, you can release a few more high energy electrons and store them in NADH. So that is the end of the Krebs cycle. Let me repeat, you got your acetyl group added in, joined to this 4-carbon molecule to produce a 6-carbon citric acid. Then you start to break down that 6-carbon citric acid, releasing the carbons as CO2 and getting some NADH out of that process. The 4-carbon molecule that you're left with can be rearranged into this final 4-carbon molecule, and you can get more energy out of that. So I got interrupted, I forgot where I am, but the overall point, again, of the citric acid cycle is to produce energy carriers and some ATP. Okay? And in the byproduct, you are releasing CO2 as you break down the carbon molecules that are involved in this. So moving on to the electron transport chain, or actually, sorry, here are some more complex versions of the Krebs cycle. You can see all of the little intermediates and at least see that there is a difference between the 4-carbon molecule that was here and the 4-carbon molecule that ends up here at the beginning. So you can see some rearranging of different bonds and things like that that would release some more energy. Okay, and there's a good animation in the notes as well. Okay, moving on to the electron transport chain. This should be pretty easy because you've studied an electron transport chain before, and it has a lot of similarities to the one in photosynthesis. So the location is the inner mitochondrial membrane. It's got to be in a membrane because we got to have those membrane proteins that the electrons are bouncing around. The description is that you're going to use those electron carriers from the Krebs cycle to produce ATP. So if we look at the details here, so some differences between the photosynthesis one is that in this case, the electrons that are powering the electron transport chain are coming from the electron carriers. They're not coming from water. So these electron carriers deliver high energy electrons to the electron transport chain. Just like in photosynthesis, as those electrons bounce around through the membrane proteins, you are pumping hydrogens, in this case, into the inner membrane space, and you're building up a concentration gradient of those hydrogens. Let me put the information up here. So first, the electron carriers pass electrons from protein to protein in the inner membrane of the mitochondria. This movement pumps the hydrogen ions or the protons into the inner membrane space. And then you have forgot about that. So at the end of this, the electrons have to go somewhere. They can't just keep bouncing around through various proteins. So in this case, the electrons are going to be stored in water. So this is another difference with photosynthesis. Instead of the electrons starting off from water, now you're going to store them in water. So that's why you need the oxygen for this process. So the oxygen is going to grab those now low energy electrons. It's going to grab some hydrogens, and you're going to end up with some water being produced. Okay. And then finally, you can produce the ATP. So the chemiosmosis, where the hydrogens diffuse back into the matrix from high to low concentration, and that movement through the ATP synthase molecule will cause the spinning, and we'll use that spinning energy to produce ATP, to add a phosphate to ADP to produce ATP. Okay. So, and there's the details of that process. So in other words, the electron transport chain, the whole point of this is to produce ATP, just like the one in photosynthesis. And it's going to do that by powering the movement of hydrogens that are then going to diffuse back through the membrane. Okay. Here's another version of it. One thing I want to point out is that notice that the NADH does start off with higher energy electrons, so they start off earlier in this process. The FADH starts off a little bit later, so they end up only pumping a couple of hydrogens, less than the NADH. So in other words, they can produce less ATP and power the overall process. Okay. So again, very similar to the one in photosynthesis. The difference is really being the location and the source versus where they end up of the electrons. Okay. So our overall energy output for aerobic respiration is summarized here. You end up with 2 from glycolysis, a net gain of 2, remember. You get 2 from the Krebs cycle, and then you can get 32 ATP from the electron transport chain for a total of 36 ATP total for each glucose molecule. Now, sometimes you'll see this number slightly different, like maybe 34 or 38, and that just depends on if you like count all of the ATP from glycolysis or not, or a couple of other different factors, but it's going to be somewhere around 36. I would never ask you if it was 34 or 36 or 38. I would only put in like 36 versus, I don't know, like 20 or something like that on a test. Okay, and this diagram also summarizes the whole process. So glycolysis, 2 ATP in, but 4 out, so 2 net. The pyruvate moves on to the Krebs cycle. From that, we get all these electron carriers that go into the electron transport chain. We also get the 2 ATP here, and then overall, you can end up with 32 ATP from the electron transport chain for a total of 36 ATP. Okay. And this is another good diagram that kind of summarizes the entire process. So if you get confused, go back to the big picture, and this is a really good one to show what's happening in the process. Okay, so there's our summary of